So like Ken was saying, I'm Ishika Kumbhakar. I'm a master's student, originally from upstate New York. I did my undergrad in North Carolina at UNC Chapel Hill and studied geography and environmental science. So I don't really have a background in botany or plant pathology, but this project applies the background that I have in geography to help solve applied problems in plant pathology. And specifically, this project will be looking at crops in rotation with potato that predict the occurrence of mefenoxam-resistant Pythium species. So my research here revolves around potatoes. The United States is the fifth largest potato producer globally, with potatoes being the top vegetable crop in the nation. And out of that, 57% of production occurs in the Pacific Northwest. The Pacific Northwest is good for potato production for multiple reasons. It's got a long growing season, good light intensity during that growing season, sandy soil with good drainage, warm days with cool nights, and access to water from major rivers. So one major disease in potatoes is Pythium leak. Pythium are soil-borne pathogens in the class Omycota, and they have a very wide host range, and oospores can survive in soil for multiple years. And oospores, which are the overwintering bodies, shown on the lower right here. And this disease causes leak or water rot in potatoes, and that's characterized by a rot of the interior of the tuber, which sometimes creates cavities. And if you look on the lower left picture on this slide, you can see what a really bad case of leak might look like, with that brown rot and a dark brown delineation between diseased and not diseased, or not non-symptomatic potato. Another symptom is if you cut open a diseased potato and squeeze it a bit, some liquid will drip out. One common management practice for crops is rotation. Alternation between different crop types and leaving land fallow has been practiced since early in human history or agricultural history to reduce soil depletion. So crop rotation, it was mentioned in the Bible, it was common during the Roman Empire. It's been around for a long time. And it's effective because it interrupts the pathogen life cycle in terms of time and space. And so rotation is one method of controlling soil-borne diseases. But there are some things that can go wrong with soil rotation. It's not a cure-all. Otherwise, our lives would be very easy. Pathogens like Pythium have a broad host range. And so rotating doesn't necessarily break up the life cycle in terms of time. Additionally, if pathogens are exposed to the same pesticide during a rotation, they may end up increasing in their resistance to that pesticide. Another problem is that there's been growing demand for potatoes, and to meet that, rotation length could be shortened. However, this risks increasing disease and reducing productivity. Crop histories and associated pest management practices can influence pest biology. However, management in the form of pesticides can actually be detrimental in the long term. So there's a little graphic on the right-hand side here that shows how pesticide resistance might form. Repeated exposure may result in selection for individuals that are able to metabolize a pesticide. And even if a grower is rotating crops, they might still be using the same pesticide or fungicide on multiple crops within that rotation. So over time, this can result in quality reduction and crop loss. One type of pesticide that's commonly used for potato crops is the fungicide mefenoxam. Mefenoxam it's the active ingredient in Ridamil, and it's a systemic pesticide used to control oomycetes. Because it's broad spectrum, it's often used for potato as well as other crops commonly found in potato rotations. Due to this high rate of use, resistance to mefenoxam has evolved in several important potato pathogens, including Pythium ultimum. So here's an example of 11-year rotation for 4 fields. And because Pythium has such a wide host range, it's possible that pretty much all of these crop types could be affected by it, or that they could be hosts. And out of the crops shown, the ones that are in green are crops that mefenoxam could be applied to. So that's onions, potatoes, alfalfa, and peas. And that's just out of this example. There's a really high number of crops that mefenoxam is applicable to. So for my research, I have the following 4 objectives. First, I will be using USDA land cover data to determine crop histories. Next, I'll develop a crop production matrix for crops commonly found in rotation with potato. After that, I'll use crop history and estimates of mefenoxam exposure as predictors of mefenoxam resistance development. And lastly, I will validate the prediction model by sampling fields and assaying for mefenoxam-resistant Pythium isolates. So to reach my first objective, I'm working with CDL data, and that is a crop data layer that's created annually by the USDA via remote sensing. So essentially, they're taking satellite imagery and classifying the landscape based on what's growing there. The result is a pixel map, and in the current raster or pixel form, the data is not all that useful to us because we need to define the field management units to answer our questions. So the first task involves defining field boundaries and setting up these field management units. So what we have here are 2 fields overlaid on a landscape map, and we can see what pixels are there. Using this identify tool, we can see that there's alfalfa on the left-hand field and potatoes on the right. But to determine the actual field management units, we need to draw boundaries, which is called digitizing in the GIS world. When we're doing that, we see misclassifications and imperfections if you look along especially along the borders of these fields. And a lot of that is due to resolution. The CDL data is at a resolution of 30 meters squared. So when you get to the edges on a finer scale, it's going to be pretty chunky. But our goal here is to determine majority pixels so we can identify what crop has been grown in this field for a single year. In order to determine which fields need to be digitized, we want to know which fields have grown potatoes in the past decade. This means essentially sandwiching all of that CDL data for each year together into a file that combines all those years and digitizing based off of that information. So this here is a heat map I made that shows the frequency of years potatoes were grown in the respective fields for this farm. And visually, we can see that a lot of the fields had 1 to 3 years of potato growth, and the maximum was 4. So the previous slide was just a small subset of what I'm doing. That was about 300 plus fields. And I want to do the same task, but I'm doing it for all of the fields in the Pacific Northwest, or all of the potato fields. And that's going to be a low estimate is 8,000, still working on it. And that's for all of the years that the CDL is available. So from 2008 to 2019. And this map shows potato pixel frequency in the same manner as the previous heat map. It's just not fully digitized yet. And just for reference, on the upper left, we have the Columbia River Valley, where Oregon and Washington. And on the lower right is mainly Idaho along the Snake River Valley. And to reiterate, we're not just getting the year that the fields have had potatoes grown in them. We're getting the entire crop history, which is what we need. So between CDL data and grower records for about 12,000 field year combinations, we found an agreement of around 88% right out of the box, and that was improved with some adjustments. One adjustment that we made was from just an herb classification in the CDL data to mint classification in some cases. And that was based on grower records, so we could get a little more specific and accurate in our crop data. And another problem that we had, or one of the problems that we had, was distinguishing between winter and spring wheat, where grower records that we received didn't contain that information. And it probably exists, but in this preliminary check, the agreement was improved by just not distinguishing between which season the wheat was grown in. And so we could improve that to about 92%. And one thing to note is that classifications of crop type are likely getting better as time progresses. We're going from 2008 to 2019. That's a pretty long time in terms of technological advancement. Our next goal is to create a crop production matrix. To do this, we will extract crop information from each year and determine the crops most often grown in rotation with potato. So we're doing crops most often grown, and that makes it easier to pare the problem down to something manageable and will likely represent the greatest number of acres per grower rotations. And additionally, some types of crops are likely grown by only a few growers and wouldn't be as applicable for this wide-scale study. So for these crops, we want to determine application rates of mefenoxam and compile those rates to find full exposure per field over the time period. And that third point will be a challenge. Determining the rates is going to be one of the harder parts of this project. And some of the things that we've been considering have been surveys to growers, looking at labels and suggested application rates, as well as extension recommendations for growers. So this is an example of a production matrix. The information is appended to the crop name in the rotation matrix, and that will allow for the derivation of total production practices specific to a field. In this matrix, we've included insecticide, fungicide, tillage, and irrigation. So there's the potential in future study for not just looking at one aspect being mefenoxam exposure, but you could also look at other management practices. So this would be an example of a matrix where we're just looking at pesticide application, and that allows us to explore more of the possible predictors in a simplified way. You can see here that all of the fields have the same total exposure, but we're ignoring for crop type and other management practices. So the question that we would pose here is, which field is most likely to develop pesticide resistance? Does annual exposure matter or not? Our third objective is to create a predictive model. And we've created a preliminary data model to predict the presence or absence of pesticide-resistant pathogens, specifically Pythium, based on crop histories and mefenoxam use. And this was based on around 35 fields in which soil plating was done. And the model was done using multiple regression methods, specifically best subsets. It's pretty interesting that even in a small data set, we found that sweet corn, peas, and field corn were predictors of mefenoxam resistance. So once this model has been expanded and developed more, we want to confirm our model. And we're not here yet, but we want to identify fields with rotations correlated to pesticide resistance, and then randomly select sample fields and compare to our predictions of resistant versus not resistant. And this is something that would potentially happen next spring or summer. So just to wrap things up, rotation information can potentially help us predict fungicide-resistant pathogen occurrence, and that's the basis for this project. And things are looking good so far with even the test data set. We found some interesting results between crop histories and mefenoxam resistance. So I'd like to quickly thank the following people and sources of funding. Everyone in the Frost Lab, Anders Huseth from NC State, who introduced me to this project and got me out here. And funding-wise, the Oregon Potato Commission, Northwest Potato Research Consortium, and of course, Oregon State University. And now I'll take any questions you might have.
All right, Ish, thank you so much. Do we have questions for Ish? All right, Jay.
Yeah, I'm kind of curious. It's a very interesting study. Can you use elevation data to try to figure out maybe low areas of the field where you might be likely to find more Pythium or maybe more resistance to mefenoxam? I'm just trying to, how do you take this 120-acre field and take a sample?
Right. So that's something that I've considered. But that's, I mean, the scale of the project that I'm looking at now is on the range of over 8,000 fields. So you could do like a per field study, but that would really be another entire project, I think, just because there's likely so much variation within a field. But I think to account for that, the best thing to do would just be doing a random sample of fields, potentially doing more than one sample per field, something like that.
John?
So Ish, given your the results you had, the preliminary results, sort of suggesting this linkage between the crop history and, I mean, do you care to speculate on any possible underlying reasons for that in your data set?
Well,
hypotheses for sort of future testing.
Yeah, there's a lot of research still to be done.
Sure.
So I wouldn't be confident in making some conjectures quite yet.
Okay, fine.
Michelle?
This is from Gabe. One question I had was, if different crops had different fungicides registered for use, could it be that the crop history has an effect only because of the fungicides that are registered and not because of the crop itself?
Yeah, that's a possibility. And that's, so for this project, the the matrix that we're looking at would be a combination of the crop and the fungicide being applied. But are you asking like if it's not mefenoxam, if it's something else?
Well, you mentioned, for example, that sweet corn and peas had an effect, but only peas had mefenoxam registered for use. So I guess it's a little building on John's question, but if you grow more peas or onions, which you could use mefenoxam on, it seems like that would be the reason why you'd get a resistant isolate and not maybe and not the crop itself.
Yeah, so in this, I guess we're looking at both things. We want to look at both the rates of pesticide being applied and the crops that are being grown.
Okay, thank you.
Melody?
Yeah, I was wondering, and I may have missed this, what level of resistance are you evaluating? Because there are different levels.
So I haven't actually done any of the resistance testing. All I know is that it's measured by culturing for Pythium, and then or like culturing in presence or absence of mefenoxam. So I'm not sure to what level of resistance the isolates are being measured to.
Oh, okay. Thank you.
All right. Any other questions for Ish? All right. Seeing none, let's unmic and thank Ish.